The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, and thank you for joining us today. You've logged on to the Nelson Labs webinar, Cleaning Evaluations of Newly Manufactured Devices. Just a few housekeeping items I'd like to take care of before we get started. If you missed one of our webinars or simply like to refer back to one, you can always find them on the Nelson Labs website on the on-demand webinars page listed under Education. You can also receive notifications of upcoming live and on-demand webinars by liking Nelson Labs on Facebook or following us on Twitter or LinkedIn. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. So let's get started. Today we're joined by Aaron Bakes, Associate Technical Consultant in the Expert Advisory Services Department. Aaron is she's been with Nelson Labs and has been a part of the medical device industry for over 17 years. Prior to her role in consulting, Aaron was a department manager for the Nelson Labs Chemistry Group, where she oversaw various analytical test methods, method development, and equipment validations. Aaron joined the Technical Advisory Group in 2017 as a technical consultant for cleaning validations for newly manufactured devices and chemical characterizations with extractable and leachables analysis. Aaron is a member of the ASTM F04 Technical Committee for Medical and Surgical Materials and Devices. She also has participated on the ISO TC 150 Working Group for 19227 implants for surgery, cleanliness of orthopedic implants, and general requirements. We do encourage your questions, and you can submit them at any time in the questions box. Aaron will try to answer as many as she can in the last 15 minutes of this presentation. And now to begin, I'd like to turn the time over to Aaron.
Great, thank you, Mike. Welcome everyone, and thank you for joining. As Mike said, my name is Aaron, and I'm going to go over some general concepts for cleaning validations of newly manufactured or single-use devices. So let's get started. We have a lot to cover. When evaluating medical devices for safety purposes, I think many of us are pretty familiar with sterility and biocompatibility programs, at least as far as the necessity of each and the regulatory expectation for a successful assessment. But there is still often confusion on the role and the value of a cleanliness assessment. Because if it passes current biocompatibility requirements, then the device is clean enough. However, cleaning validations are ideally performed upstream in the device lifecycle, after the processing and cleaning programs are finalized, but prior to biocompatibility. Completing a successful cleaning validation may help prevent issues later on in the much more expensive and time-consuming biocompatibility. Also, biocompatibility testing is expected to be performed the one time unless there are changes to the device. Unlike cleanliness assessments that are also expected to be used in routine monitoring programs and part of GMP 21 CFR 820 production and processing requirements. So before we get into validation development, I wanted to just give a quick history check that many of you are probably already familiar with, but just in case. One of the more famous cases which illustrated the gap cleaning validations would cover was a situation with orthopedic implantable devices. So during the manufacture of acetabular cups, oil was actually introduced to the device. And I don't know if the oil was not noticed or if it was just deemed a low risk because at the time the biocompatibility had actually met requirements for that time. But these high residue devices were implanted into thousands of patients. And unfortunately, residue prevented successful osseointegration and caused inflammatory reactions, which then led to thousands and thousands of recalls. So that company, of course, went out of business, and many people are still not walking today. It just really emphasized the necessity to monitor and ensure that the manufacturing steps are in control. And I mean, there have been quite a few lessons learned since then. It's certainly more common to for customers downstream to require their component or device suppliers to complete cleaning validations on their phase of the device manufacturing process. So now, while the Sulcer event, of course, acted as a catalyst or a driver for enforcing cleaning validations, biocompatibility also did evolve as well. I mean, with the development, the further development of isochemical characterization, including extractable and leachable programs, this type of situation is really highly unlikely to slip past a toxicological assessment today. But something interesting, there is a significant argument for the safety of regularly used medical grade materials, like metal materials, like stainless steel and metal alloys. You know, at this point, we're pretty confident in the biocompatibility of these materials, and it's actually the surface residue from the manufacturing of the materials into the final finished form of the device that's the real concern at this point. So subsequently, we've seen cleaning validations for ISO 19227 accepted in place of extractables and leachables in just certain situations like change assessments of these types of non-polymer materials. Again, just really emphasizing the value of a proper cleaning validation. So now let's get into the development of a cleaning validation protocol. One of the first steps in the risk assessment of a device to identify potential residues which may be found on the device. So, you know, what type of chemicals are used? Are they directly or indirectly? What type of debris could be produced? That type of thing. Once this list of potential contaminants is available, we can then categorize the contact agents and select an appropriate test method and acceptance criteria. So we're going to break this down in a step-by-step format. ASTM offers a guideline for developing this validation within the standard F3127. This is a full scope of considerations for a cleaning validation. It's not a specific outline of what to do, you know, a checkbox style. It's more of a roadmap of what to think about, where to go, that type of thing. But it's not specific to one device type. So it is really useful to walk through this standard and understand the questions and points brought up. Alternatively, ISO 19227, published in 2018, it does offer a more defined and specific outline. This is specific and intended for orthopedic implants, but in my opinion, the test strategy within is a comprehensive and non-specific approach that could be easily applied to many devices outside of the scope. So this is an overview of the validation process, and we'll go through these steps individually. But again, it starts with a risk assessment of the device and the processes, which leads to the test selection, sampling plan, and acceptance criteria. Then you can move into the testing phase and finally summarize those results with a conclusion statement relative to the efficacy of the cleaning process. Each of those steps should be documented, which is something we love to help with, and we do offer through our technical consulting services. So once you have the processing steps identified, and here's some examples of what that could look like. I mean, obviously there's going to be quite a bit of variety out there with respect to different medical devices, but these are some common steps we see. 3D laser sintering, 3D printing, additive manufacturing it's a pretty challenging step in a cleaning program. That residue printing powder can remain behind in the porous structure of the device, in the layers. They might be heavier particles that are difficult to extract with standard extraction parameters. So it's certainly a challenge to keep in mind. machining, that could mean a variety of things, and it could introduce a variety of potential contaminants, coolants, lubricants, acids, all types of things. You might have an in-process clean with possibly house or city feed water. Ideally not, but maybe. You might have cleaning agents, more aggressive cleaning agents at this stage, and solvents, degreasers, that type of thing, all kinds of variety as well. And then a final clean, again, possibly more detergents and cleaning agents, hopefully DI water at this phase, but maybe not. Maybe there's a city feed water at this phase, and maybe a solvent rinse like IPA, that's possible. So then you have your steps identified and those contaminants or potential contaminants that you're introducing to the device, and maybe contaminants that are produced through the manufacturing process. You have those identified. Now it's a good idea to familiarize yourself with potential classifications. This will help with those test strategy choices you'll need to make. So water soluble, that's a very broad category. It's going to include a lot of different chemicals. Detergents, metals is listed under water soluble. There are water soluble metals in maybe city feed water, something like that. Different types of water soluble solvents, polar organics, polar inorganics could be listed on there, nitric acid or citric acid, sodium hydroxide, all kinds of things under the water soluble category. The water insoluble category might be a little confusing, but it's kind of two subcategories of heavy oils and particulates. Particulates is actually extracted with water, but we're talking about that particulate debris, like metal flakes, the 3D printing powder, polymer flakes, fibers, all kinds of things could fall under that particulate category. And then biological, living and dead, primarily talking about bioburden and endotoxins. So to categorize those chemicals, it might be pretty obvious, and it might be really easy to say this is an oil, it's going in that non-polar category, that water insoluble category. But if you're not as familiar with the chemical or the chemical properties, you might have to do a little bit more research. The first step is usually an SDS, which, if you're familiar with SDSs, you know they can be terrible. They can be not complete. They might lack consistency. They're not regulated. So sometimes it's missing information, sometimes it's not great information, but sometimes it does the job. If it doesn't, and you need to dig a little deeper, there are options available online, better options, I think, like PubChem. We've got an example of IPA and what you can look at through this website. You can look at chemical and physical properties. You might want to look into the toxicity of the chemical. We'll talk about that later. So this is a great option. It is government curated. It is a little more reliable. It's a lot more reliable. So it's very helpful. Another example is the EPA chemistry dashboard. Again, great information on different compounds out there. So now that you have walked through all your research, identified your potential contaminants, and categorized those contaminants according to solubility, the next step is test selection. There are all kinds of choices which could be made at this point. We're going to stick pretty closely to the outline in ISO 19227. This is primarily a non-specific and quantitative approach, like NBR, TOC, and THC, which we'll get into. And it matches the typical recommendations that we've been making for many, many years. But it's worth noting that ISO 19227 is not absolutely strict with test selection. It emphasizes a risk-based approach and allows for alternative test method selections with justification. So, for example, perhaps you decide you do want to run testing with specificity. You might choose a method like GCMS, which is gas chromatography, which will give you a quantitative mass of residue as well as compound identification. This is something that would be expected in an extractable and leachable program per ISO 10993-18. So of course, it's going to be acceptable for a cleaning validation. It just might be overkill. It may not be necessary. So it just is going to kind of depend on your specific situation. But in general, non-specific quantitative methods have been successful with cleaning validations. Also, because of the higher cost and the higher turnaround time of testing with specificity like GCMS, it's not really ideal for routine monitoring as you incorporate that into your program. So looking at this table, kind of from the left to right at the bottom, you can see gravimetric non-volatile residue and TOC, both under the water-soluble category. And we're going to dig into these a little bit more on later slides, but the gravimetric non-volatile residue method is strictly non-volatiles. It does have an evaporation step involved. So any semi-volatiles like IPA is going to be lost in that process. But it's not specific to organic or inorganic. If there is, sodium hydroxide or nitric acid on your device, that's going to be extracted and detected or in that measurement, that non-specific measurement, as well as detergent residuals, as well as if you have metal flakes on the device, that's also going to come off in the gravimetric water extraction. So it's a broad scope of potential residue, which has pros and cons, right? Versus total organic carbon, TOC. This is another water extraction. It is not specific to non-volatiles. So if there's IPA maybe in a blind hole of your device or a textured area that just kind of got stuck behind, TOC would detect that as a little more volatile. But it is specific to organics. If you have inorganics on your device, TOC is not going to catch that. You'd have to go back to gravimetric or another method. So moving over to the water insoluble side, here we've broken out particulates and heavy metals separately to give a little more clarification there. Heavy oils, just pretty straightforward. If you have oils on your device, you're going to need to extract in a non-polar solvent, such as hexane. That whole idea of like dissolves like. Heavy oils is not going to be extracted efficiently with a water extraction. And then you can use a method, either gravimetric or THC, total hydrocarbon. Total hydrocarbon is kind of the other side of the coin to TOC. We would extract in hexane and then pull an aliquot and put that on actually a GCFID to give a quantitative amount of non-polar organic carbon. If your device, this is great for all metal devices. Hexane is compatible with metal and THC has a nice sensitivity. It's also listed in ISO 19227. If your device has polymer components, it's not going to work well with that hexane. If you're working with another THC method that does offer alternative non-polar solvent extraction options, like maybe it extracts an IPA and then goes on the analyzer, that might work for you. But our method at this time is just hexane only, so it's not going to be compatible with polymer components, in which case we need to switch it over to the gravimetric method to detect those oils. So kind of backtracking a little bit to the particulates and the metals. This column can get a little complicated. So USP 788 is a particulate method. It's, we would extract in water, and that extract would go on a light obscuration particle counter to give you a count of particles. Could be metal particles, could be fibers, could be polymer, it could be any number of things. And there is a microscope option instead of, or in addition to the light obscuration particle counter, which gives just a little more qualitative information. The other option is NVR. We kind of talked about that earlier with the water soluble column. Gravimetric will pull off those particles as well. So if you have a device and it has detergent on it, and it also has metal particles, then that's both going to come off in the gravimetric extraction and just kind of be one lump sum of mass. With the USP 788, that's going to give you a count of particles based off of particle size of those metal particulates. The last option, ICP for metals is where it's great information, but again, it's a little tricky. So let's say your device has been exposed to city feed water and there was heavy metals in that water, and that's kind of remained behind. Or there was some odd solvent or something that's had heavy metals in there. If it's water soluble, the ICP method will extract it and detect it. If it is not water soluble, like metal flakes, the ICP method can extract it and detect it, but it needs additional preparation steps that are not routine, like an acid digestion or a microwave digestion. It needs to get those metal particles into solution so it can be analyzed on the ICP. So when we say ICP for the purposes of cleaning validations, we're usually just talking about water-soluble metals. And that's going to give you great data. It's going to take a little bit longer than like the gravimetric method. And then kind of moving farther to the right, we have biological, which again, we're talking about BET, bacterial endotoxins, bioburden, and cytotoxicity. So let's get into these again, where we're kind of just re-summarize. So the gravimetric method, we'll take the device, immerse it in a solvent. It can be it can be water for the polar solvent, or it can be non-polar solvent. There's a few choices, and we just choose the one that's most compatible with the device material. So you can send 2 separate sets of samples, one for the water extraction, one for the non-polar extraction, or if you're just opting for one set of samples. We extract it. Typically, our extraction is for gravimetric TOC, THC, is a 30-minute sonication at room temperature. This is per ASTM F2459, which is the gravimetric standard that allows for those extraction parameters. ISO 19227 lists this ASTM F2459 standard, as well as ISO 10993-12, which is kind of the regular biocompatibility extraction part, as extraction options for cleaning validations. So we like to use the 30-minute sonication because we want to be as aggressive as possible at removing that surface residue without degrading the device. We're not directly concerned with the clinical relevance because we're also talking about just the efficacy of the cleaning steps. So we want to be aggressive in the extraction. A 30-minute sonication in an appropriate solvent is pretty aggressive. But it's all going to come down to a recovery efficiency. I should back up and continue the remainder of the analysis here. So after the 30-minute sonication, we let that solvent evaporate all the way off. So we're left with the solid residue that we've pulled from the device. And that's what we take the mass of and report. So it's just a non-specific lump of residue, a microgram per device. It's a more sensitive test method than you would expect due to how we've improved techniques over time and how we've been able to control the background noise. We have pretty good sensitivity for an NBR test. It's pretty comparable to TOC, not as sensitive as THC, definitely not as sensitive as a GCMS. It does require a recovery efficiency. So the method itself is validated, at least at Nelson. It should be validated at whatever lab you choose. But the gravimetric, the TOC, the THC methods, these are methods that are validated, but we can't validate every type of device out there with all the different residue possibilities out there. So that's why these recovery efficiencies are necessary. Recovery efficiencies will demonstrate that we are able to extract the residue from the device with the given extraction parameters in the solvent and detect it with the appropriate test method. We want a high recovery. So ideally that's performed on a dirty device, because if it's a clean device, it's hard to get a high recovery from a non-detect, right? That's a challenge. So we try to encourage dirty devices. There's a number of ways you can get those dirty devices. It just depends on the situation. Sometimes you can pull it from the line after a certain step. Sometimes you have to dope it with the chemical. or spike it. So you can send the chemical to the lab can spike it in a controlled setting. So there's options. It can be tough with certain types of manufacturing situations, but we just do the best we can to get that good recovery efficiency, which supports the data found. TOC, again, we'll just kind of re-summarize some things already talked about, but it measures the total amount of carbon extractable with water. So if there's that IPA, it'll go into that water extraction. Again, it's a 30-minute sonication, and then instead of evaporating, we pull off an aliquot and put it right on a TOC analyzer that produces that carbon microgram per device. Well, it'll be a microgram per mil, and then the lab will calculate it into micrograms per device, or per surface area, if that's preferred. Like gravimetric, it's non-specific. It's a water-only extraction. It has very nice sensitivity, and a recovery efficiency is also required here. Just once per device type and per method, and then it can kind of be held on file if more testing is needed at a later point. And then again, there's not a slide for THC. So just as a reminder, THC is the flip side of TOC. THC is extracted in a non-polar solvent to detect primarily oils. At Nelson, it's a hexane-only extraction. An aliquot is put on a GCFID for analysis. Great sensitivity, and the result again is a microgram per device. And a recovery efficiency is also needed for THC. Particulate matter. Particulate matter, this is the test that again overlaps with gravimetric water. This will give you a count of insoluble debris or particles extracted in water. It's non-specific. It's a shorter sonication or a manual shake or an orbital shake, depending on the specific device, in low particle water. Those particles are going to go into solution pretty easily. We don't have to have an aggressive extraction for this method. We use a light obscuration particle counter, but there is a microscope option available for additional qualitative information. And we typically measure in 2 particle sizes, but other particle sizes are also available. And then the standard we comply with is USP 788, particulate matter for injections. I also don't have a slide for ICP. I don't have a slide for every test method because there's just too many that are possible. It might get a bit overwhelming, but ICP is the other method to keep in mind. If you choose to, if you want that testing with specificity, if you want results of metal elements with an identification of the metal element as well as a quantitative mass. It probably either a 30-minute sonication or an ISO 10993-12 extraction, just like TOC and gravimetric options. An aliquot would go on the ICP-MS for analysis to give you either if there's just one metal element you're concerned with, or maybe 2, we could run the report that way. Otherwise, if there's multiples or if there's unknowns, we would just do a full screen of 46 elements. And this data is data that can be directly assessed by a toxicologist, so it's very straightforward and easy to handle, easy to address, but it takes longer and it is a bit more costly. So those are just 2 things you'd want to keep in mind when making your test choices. Then moving forward to that biological side, bioburden, we're going to measure the amount of viable organisms on a device, extracted in tween, filtered, incubated, and then the colony forming units, the CFUs, are counted and reported. Like the other test methods, gravimetric, TOC, THC, recovery efficiencies are also required here. But unlike spiking with a chemical, the lab would be inoculating, or you could provide a device with that's expected to have a high bioburden count on it, natural bioburden. These, for the purposes of a cleaning validation, bioburden is performed on non-sterile devices. We don't want these sterilized. This is not intended to substitute as a sterilization validation. It's really just intended to ensure that coming out of your final clean, your devices do not have such an outrageous organism count that it would later impede a sterilization process. So we do want these non-sterile. Next up is bacterial endotoxins. We measure this using a turbidimetric method, extracted in water for USP 85 and AMI ST72. This does have industry acceptance criteria. Side note, endotoxins are usually or often introduced through water sources. So the cleaning step itself might be introducing endotoxins to your device, which are very difficult to remove. It does not equate to sterilization. They're two separate ideas that often get confused. Sterile and endotoxins are not a concern. You can send your devices non-sterile to be tested. You can send them to sterile to be tested for endotoxins. If your device has lymphatic fluid or blood contact or cerebral spinal fluid contact, you're really going to want to look into monitoring your endotoxin levels. And now we're going to take a little break for some bad jokes. Why did the crab cross the road? It never did. It used the sidewalk. And why don't crabs donate to charity? Because they are shellfish. So now we're funnier. Okay, back to the test strategy. You've identified your manufacturing steps and the potential contaminants introduced or produced by those steps. You've categorized your contaminants so that you can find an appropriate test method. And here's just some examples. That 3D printing, that's going to produce particles you're definitely going to want a particulate test for. Coolants, they can actually be water soluble or non-water soluble. Usually they're non-water soluble, so a THC or gravimetric method, or maybe a GCMS method, but probably more often than not, it would be THC or gravimetric. Cleaning steps, house water, that's going to introduce the bioburden, the BET, the TOC. Maybe you'll still want a gravimetric. So these are just some different options to examples of what to do here. You know what? There wasn't a slide for cytotoxicity. I do want to emphasize cytotoxicity. I know we talk about this a little bit later in the presentation, but I'm going to I'm going to skip back. I'm going to skip back here. Cytotoxicity is a test I would always include in the cleaning validation. It's per ISO 10993-5, and it does have scoring criteria for acceptance in this standard. So, 10, 15 years ago, maybe somebody would have thought cytotoxicity would just stand on its own. They just lean on that to show that their cleaning process works. We know now that it doesn't stand on its own. A full risk assessment is needed. A full assessment of quantitative residue is needed, but it is very, very supportive. A good, successful cytotoxicity test is very supportive of the overall justification. So I would always include a cytotoxicity test unless you have a good reason not to. So there's your test strategy. The next big step is to get into sampling plans and replicates. I'm going to take a little sip of water here. Okay. So with sampling plans, the first step is to try to group similar devices whenever possible. Devices that are coming from the same suppliers, manufactured in the same locations, using equivalent processes, manufacturing and cleaning processes and equivalent chemical agents and contact agents, devices with equivalent or worst case materials. We want to group those together. And then kind of the last step tends to be geometries and surface areas. If you've established a group with all of those equivalencies, and then you're just looking at which is going to have the most challenging cleanability based off of geometry, you can group some more. So in this example, we have 4 plates of different sizes, as well as 5 different screws. Some might argue that if they were produced with the same materials and the same processes, that the screws would be a more worst case cleanability, and so they might pick a representative test device from the screw grouping. But if you want to just be a little more conservative, you might pick a plate as well as a screw to evaluate. And then within that group, probably just the largest surface area, so we can get better test method sensitivities. You know, with TOC, with gravimetric, THC, it tends to be the more surface area, the better the sensitivity of the test. So if you have a larger one, that's what we're going to try to push for. Sometimes with sample availability, you know, maybe with this plate set, you might actually offer the 2 largest. I don't think that's the end of the world. So again, in this example, we separated those into 2 different families, took the largest size from each. There, in this example, we separated titanium from stainless steel. Again, you know, some might argue that that could be grouped due to cleanability of the device and the material. You might go back to the individual manufacturing processes and just see if one is more worst case than the other. And then if you have 2 different devices that are different colors just due to anodization, I would certainly group those together. Test quantities. This is always a big question. So if you are familiar with the cleaning validation standards, you might have noticed that they're quite vague with respect to test quantities. A good cleaning validation should demonstrate process repeatability through separate lots, separate batches. There's a number of ways you could define that, but you want to show that your process is consistent from lot to lot or batch to batch. The standards might make a vague statement like statistically justifiable. If you're familiar with statistics, you know that that could mean that could quickly add up to a pretty large number, just depending on what kind of confidence interval you're looking for. More often, I mean, that might be 30 plus devices per device type and per test. So it can be pretty high. And I have seen that done. I have seen that included in cleaning validation plans. More often than not, I see a three devices because due to the burdensome nature of acquiring that many devices. So most often I see 3 lots with 1 device per lot to produce 3 data points per test method and per device grouping. I have seen a bit of a push lately to increase that on higher risk devices like implants. Sometimes that's just 3 lots of 3 for a total of 9 data points. Again, in the statistics world, that's not a huge difference, but it's sort of theoretical versus reality. And with reality, you're sort of doing the best you can. Randomly, I saw pushback where 15 was encouraged. There wasn't really a reason given behind that, but it happened. So if you have a higher risk device or you want just a little more confidence, you might increase that number from 3 lots of 1 up to whatever is reasonable. Something to keep in mind, depending on the lab you're testing at, you're probably going to have to produce or provide separate sets of samples for each test method. So if you're talking about 6 or 7 test methods with 3 lots, it can get quite high. Then you add on to that, if your device is very small, if it's not going to meet the test method sensitivity requirements or recommendations, you might have to pool multiple devices together to bring up that total surface area per sample. So I've seen plans where that weren't, 30 samples per grouping, but were just kind of a reasonable 3 or 9 amount, but they still required hundreds of samples. So that is something to keep in mind when you're starting off on your cleaning validation plan, that you might need a high quantity of devices available. And sometimes that even affects the test strategy choices. But it is a pretty important consideration. And then that leads us to the $1 million question. How clean is clean enough? Acceptance criteria. The good news is, well, I'll start with the bad news. Bad news is there's not a standard that says exactly what criteria is acceptable for all of these test methods for a cleaning validation of a medical device. But the good news is there has been a lot of progress. ISO 19227 that published in 2018 actually put numbers on TOC and THC as starting points, 0.5 milligrams per implant, per orthopedic implant, granted. USP 788 particulates does offer limits for water for injectables. Not medical devices. It's not designed for medical devices, but it's sort of the best we have available. And the argument is, you know, if it's conservative enough for water for injectable that can be introduced to, you know, circulatory systems, then it's conservative enough for a medical device, or it's more conservative, you know. And then VET USP criteria is also available. So you're not always starting from scratch. However, there are alternative options. Historical data, perhaps you are not running this as kind of a new device or a new change before biocomp. Perhaps this has been on the market for a while without issue, without complaint, and you just need to establish that routine monitoring process and that trend analysis. There might be enough justification to run the testing just to establish those baselines. That's possible. Cytotoxicity is listed here, but as I stated before, I think this could be tweaked. I don't think cytotoxicity will stand on its own, but it is quite supportive. So you do a successful cytotoxicity test is a good thing. That does have acceptance criteria. And then finally, comparison to toxicological data. This is a great option, and it was used more frequently before 19227 published. So this just involves a bit more research on those chemicals found during the risk assessment of the device, and the database is available, like the PubChem and that type of thing. Finding the best available threshold, like the NOEL value, no observable adverse effect level, or low observable adverse effect level, LD50, that's lethal dosage of 50% of population. So you find the one that's the most conservative, apply the device use to that calculation and establish a threshold. If that's something you don't want to do on your own, or you don't feel comfortable doing on your own because it is significant research and involves some choices, that is something our toxicologists can help with. They're awesome and they love to do it. So there are some options with acceptance criteria, and it is an important part of your test strategy. Back to the overall process. You've performed your risk analysis. It's led to the test method selection, and you've decided on a sampling plan, and the hardest part is the acceptance criteria. It's now time to test. Ideally, you would wait until that acceptance criteria is established, that initial documentation is complete, in case your test strategy changes. So ideally, you would wait until all that is complete to start your testing, but you don't have to. There are times when overlap happens, and you can send devices in to be tested prior to that. That's your choice. But this is our recommendation, that you wait until that initial planning is complete, perform the testing, and then you're ready to compile the results and make a conclusion statement relative to the efficacy of the cleaning process. And that is it. That is the gist of a cleaning validation for newly manufactured devices. We didn't really talk about this, but in the world of cleaning validations, this term gets applied to newly manufactured and single-use devices, as well as reusable healthcare devices. So this is something probably could have better defined at the beginning of the presentation, but reusable healthcare devices falls under a totally different umbrella of standards to comply with. We see overlap between the two paths, like TOC can be used in either one, but they're quite different. So if you do have a device that is intended to be reused, intended to be cleaned in a healthcare setting or per IFU, then that does require a separate set of standards and a different validation plan. There's kind of a murky area on single-use devices that are clean for IFU in a different setting, but they're single-use, and that's it. That there's not a standard out there that specifically defines this process and tells you what to do. So it gets a bit custom. And if you run into that situation, I would just suggest calling us and setting up we'll set up a call so we can walk through it and decide on the best approach. So at this point, it's already 11:45. What remains in this presentation is a series of case studies, just examples of what a cleaning validation could look like. And we can walk through those, but I do want to make sure if there are questions, we can get through that. So maybe I'll just take 5 more minutes to kind of walk through these case studies just quite briefly. And then we'll make sure that there's time for questions if that's needed. So here we have a hip replacement. Step 1, risk analysis. There's machining, deburring, grit blasting, rough cleaning, all kinds of particulates produced through that process, multiple chemicals, a rough clean, probably a degreaser to get that off, then the final steps of laser marking and passivation, and then maybe a final clean, and that final clean could actually be introducing potential contaminants as well. So, here we go. So all kinds of options here. Once those are listed and categorized, then a test method was selected, and it looked like, in this case, NVR was used. THC could have also been used on these all metal devices. I think that would have been a good choice. BET, bioburden. And then how many replicates? Family groupings, that was also defined. 3 lots, 1 device per lot. The Thido had all passing scores. And we saw, let's see, these are, if you're talking about, to give you some context, 500 micrograms, you'd be looking at that middle column under the gravimetric if you're trying to make some kind of comparison between the ISO 19227 criteria. These look really good. These look reproducible, and they did spike recoveries for recovery efficiencies. Greater than 75% is considered successful, so they had 2 successful spike recoveries. It's pretty solid. These are successful, I would consider successful bioburden and bacterial endotoxin results. The endotoxins are all well below acceptance criteria. Bioburden acceptance criteria is one that might be established off of internal trend data, but there are other couple ways that can be used. So these look really good. There's a little bit of a spike on 10, but for a non-sterile device, that's not terrible. So here's a little highlight of the section in 19227 on that TOC and THC acceptance criteria. And case study 2, there's a liner involved. Let's see, we're doing enough time, just a couple more minutes. Here they have, this is another way to show the recovery efficiency on the left side of the screen here. The top 2 rows are the exhaustive extractions. Instead of a spike, they performed an exhaustive extraction. And then the cleans a little bit higher than the results you saw before, but still, if you were using the 19227 on this gravimetric, it would be considered a pass. Cyto looked great, bioburden looked great. So the initial validation, this is just a step of talking about routine monitoring. You might want to scale down your test plan. For routine monitoring, it just depends on the risk of your device. If you feel like you have a higher risk device that needs more close or more frequent routine monitoring versus if you want to try alternating tests or spreading out rather than quarterly, possibly, biannual or annual. So that's also based off of the lot release size. Here's some examples of routine monitoring phases and how you can evaluate that trend data by establishing those alert levels versus action levels and your corrective action plans if they exceed that. So this is a case study of a tubing set where in their routine monitoring, there was a clear trend established over a period of time. We have 5 months listed, and then they saw a spike. Obviously not expected, but it gave them cause to investigate. And they implemented an additional water rinse to flush out residual IPA, and the TOC levels went back into an acceptable range. So not all changes are beneficial. They thought they were improving the process. It turned out they were adding contamination to the device. It was getting held behind inside that tubing. And routine monitoring is what caught that issue. All right, here we are at the question portion of our presentation. Here is my contact information. I love to talk about this stuff. So I'm happy to, if you have any questions or you want to discuss anything further, please feel free to contact me. And also for more educational events and resources, you can visit nelsonlabs.com or either of our sister companies, Sterigenics or Nordion. And that concludes the presentation. So I'm going to hand it back over to Mike.
Great. Thank you, Erin, for all of your insights. And I think we've all learned some great information today, and you've given us a lot to think about. And yes, we have received several questions that on this topic. So we'll just get straight to that. We'll answer as many of these as we can in these last 10 minutes of the presentation. So, okay, here, Erin, here's your first one. Is there a limit on the length of a device for gravimetric method?
Great question. So not officially. It's dependent on the lab resources, really. If they have an extraction vessel that we can immerse the device in, then it should be fine. There are some things we can do. We can flip it. We can immerse half, sonicate, and then flip it to immerse the other half. We can, if there is just a portion of the device that needs to be assessed for some reason, which is a little tricky, but it could happen. We could just focus on that portion. And then least ideal, we can cut devices at times. If it's not a thick metal device that we can't cut through, we can. That is an option. It's just not our preference because we run the risk of introducing more contaminants. But if you run into a situation where you're just not sure, I would just take dimensions, measure some dimensions and send them to us so we can just check our current stock of glassware. We might even be able to get some special glassware for you.
Great. Thank you, Erin. Here's our next question. It looks like it might be a two-parter. Okay, the first part is, does TOC include remnants of microbiology and is therefore a predictor for bioburden? And the second part is, and does TOC include the same hydrocarbons in the total that can also be detected by THC method?
So the first part, I wouldn't plan on it. TOC will detect biological matter, but there's never been a direct correlation made. And every time I've seen a company try to, it just didn't work out. There have been official statements by the FDA regarding endotoxins that there's no correlation there that can be used. So if you have a lot of biological matter on the device, it will probably affect TOC, but it doesn't have the sensitivity enough to be used as an appropriate marker. And then the second question, probably not. There's probably not going to be a lot of overlap. The THC method is specific to C10 to C40. It's a specific range of hydrocarbons that are very non-polar. I guess it's possible. So if just in the physical agitation of the sonication, if some of those oils came off in the TOC extract and made it onto the TOC analyzer, it is oxidizable carbon. So it could be analyzed and show up under TOC, just not very consistently. if you think of a cup of water and kind of how the way the oil kind of floats to the top, on a TOC analyzer, the needle goes to the bottom of the vial and pulls from the bottom. So it's not consistent, but some could. Some overlap, nothing to depend on.
Great, thank you, Erin. Here's another question. Asking you to comment on the reason for certain metals causing cytotoxicity failures, for example, gold, silver, tin, etc.
Oh man, I think that's a better question for a biocomp expert. And we do, I see that come up with our biocomp experts from time to time, and they're just a lot better at answering that than I am. We could, if you want to send us your contact information, we could easily follow up on that and get a better answer for you. But it is related to, actually, no, I'm not going to go there. I don't want to mess that up. But send us your contact information. We'll get a better answer.
Perfect, perfect. Here's the next question. For USP-NF 2020-788, are particulates less than 10 micron ignored?
I don't think they go smaller than that. I don't think they have a smaller range than that. They tend to go higher. So maybe the concern is nanoparticles. That would show up better in gravimetric.
Great. We have time for a few more questions. Here's the next one. Can absence or let's see if I'm trying to. Can absence or too low of quantity of gram-negative bacteria be used as predictor for BET and used to rationalize omitting BET? So the question is, can absence or low quantity of gram-negative bacteria be used as a predictor for BET and then used to rationalize out of BET? And BET,
I'm not sure if
endotoxin.
I'm not sure if I fully understand that question. So if there's an expectation that there will be an absence of gram-negative bacteria, usually the idea is to test it to demonstrate with data that that's accurate. If people who label their devices pyrogen free or low endotoxin have to test it to prove that. It is the gram-negative bacteria that's getting tested. So I'm not sure if I fully understand that question. Maybe that's another follow-up if I didn't answer that clearly.
Sounds good. We have time for about 1 to 2 more questions. And here's another good one here. Can you use a full and successful cleaning validation result as an argument to skip biological testing?
Biological or biocompatibility? I'm gonna kind of assume that means,
but you can infer.
Yeah, I'm gonna assume that means biocompatibility, and that's gonna go back to your biological evaluation plan. It's kind of it's another realm of topics here, which is tough to explain in 30 seconds. But if you're in need of a biocompatibility assessment, the first step is to perform a biocompatibility evaluation plan, where you do identify the risks based off of the device use, the materials, any literature research, that type of thing. And then from that assessment, you determine an appropriate test strategy, be it biocompatibility, in vitro, in vivo stuff, or chemistry testing. So this is all like in the 1093 biocompatibility suite. There are times when with all metal devices, cleaning validation has substituted for certain chemistry testing. It's pretty limited, and I wouldn't plan on it with any polymer devices. Polymer devices are going to have leachables within the material that need to be evaluated under clinical conditions. So I wouldn't, if you have a polymer device, I wouldn't plan on any hope for a cleaning validation to be used in place of. If you have a metal device, then it's something you could look into and you could ask about. Still not a guarantee, but it's worth asking. If you do need help with BEPs, that is something our biocomp specialists are great at.
We'll go ahead and take one more question here, Aaron. For the non-organic matter, can I use conductivity testing to replace NVR method? Can you please recommend an acceptance limit for the conductivity testing of the orthopedic implants?
Yeah, I've seen people do that. I don't love it. I don't think the acceptance criteria is, I think it's trickier. You can go back to USP purified water standards and look through their definitions of and their associated acceptance criteria for ionic contaminants. And I guess there's kind of this thought of if you extract in USP purified water and you compare your extraction blank with which wouldn't have a device in the extraction, it's just basically purified water, with an extraction of a device extracted in purified water and then take the conductivity measurement of it. Maybe you can compare it to USP purified water levels, but those are volume levels. So you'd need to kind of evaluate it with respect to the device. That's why we always kind of do mass per device or mass per surface area. So if you go through USP 645 and read through that monograph, you'll see some information there. I've seen people do it. We don't usually push it. We don't usually recommend it. I just don't think it's as straightforward. And if somebody is very concerned about ionic residue and inorganics that are soluble in water and they don't want to go NVR, the other option is IC, ion chromatography. I think that's going to give you better data than conductivity reading, and a little bit more easier to evaluate acceptance criteria with the help of somebody who has that type of experience. So I see ion chromatography. It's a water extraction. It's not something we perform at Nelson Labs, but we can subcontract out, and we would have to define which ions are going to be evaluated and that type of thing. But I would maybe look into that. But that's not a hard no. That's not a hard no. You can't use conductivity. It's just not our first preference.
Great. Well, we thank you, Aaron, and thank you for all this thorough and informative presentation. I'd like to thank Erin, our presenter today, for her insights, and I'd like to thank you, the viewer, for attending this session. We hope that you found this as a valuable experience. This will be loaded to the Nelson Labs website in the coming days if you'd like to refer back to this webinar or any of our past webinars as well. Thank you and have a great rest of your day.